Here we go. So we have started this block course on developmental biology. You'll move on to maybe more neurobiology. Next week is Susan. She will probably also talk about development or about invertebrate organisms, probably about C. elegans. So in my week, in our week, we have started with genetic analysis, 3 days. And now we go to introduction to vertebrate development, 1 day, a bit more than a day and a lecture. And then tomorrow we'll discuss the papers and discuss a little bit genetics. So developmental biology is kind of all about how do animals develop. This is a C. elegans embryo. This is a Drosophila embryo. This is a zebrafish embryo. These are the adults. You see, they all look very different. Yet they seem to have a common body plan. Since about 1984, we know that all of these genes have homeotic genes. All of these organisms have homeotic genes. That came as an absolute surprise because genes that control development in flies control whether you form wings and antennae. These genes, which form either wings or antennae or legs, were conserved in all of these organisms, despite most of these organisms not having wings nor having antennae. So that was a huge surprise. This discovery was made exactly where I sit here, probably in the room beside, but that was discovered in the Gehring lab, where they cloned the first Hox genes. And then a week later, after having gone to the zoo and took other animals, found that these Hox genes are conserved. Not only Hox genes are conserved, the basic unit is conserved. All of these organisms are built up of cells. These cells have to talk to each other. They all talk to each other via the same signaling pathways, and this is very important. And so what you can do when you study either C. elegans, Drosophila, zebrafish, you can extrapolate from these studies to other organisms. You have to verify. We extrapolate from mouse to human. Now we have human tissue and organoids, and we can verify some of these things. Many, many of these basic principles of development have been discovered in invertebrates. Now the basic principles are probably out there. There are a lot of details. There are a lot of things to discover, so you can never say what is. We are there because you don't know what comes next. Here we have been talking about cell signaling the first 3 days. We didn't look at individual cells all that much. We didn't look at live imaging all that much. So what we did is try to understand how cells talk to each other. We used the DPP pathway for this. We saw that DPP can spread, so a signaling molecule can instruct other cells. Spreading is important. We use protein binders, and so maybe not as important as one initially thought. So this is early days. New methods will give rise to newer concepts. So we refine these concepts. So the molecular basis and the protein-based basis of all of these things is, of course, still under investigation. The fly, as you saw, is a very nice model system. It's a pity because you didn't really see these flies and smell the yeast and sort of get a real grab of it. That's a pity, but if you ever want to see some flies, you drop by the lab or you put some wine out on the balcony. And then the small flies that are there, these are usually Drosophila species. Yanni and Eric, they were sort of they started all of this. Use genetics for development. It wasn't clear that genetics can be used to study the very earliest things that happen in an embryo. One used genetics to look at adult flies, no wings, no legs, no eyes, double leg duplication, things like that. That one looked at. Morgan started with this, but that then you can have mutants in flies as heterozygous, cross them together and get defective embryos. There was one such mutant, and Yanni said, there must be many more. We want to find them all. And they did this famous screen. They looked at the larvae here and asked, are there mutations? You take a heterozygous fly, a male and a female, you cross them together. A quarter of the progeny will be mutant. Are there mutants that affect this body plan? There was one mutant known. They said there may be more. They developed methods to isolate these larvae easily from hundreds, thousands of strains. They collected them systematically. First chromosome, also X chromosome screen, second chromosome, third chromosome screen. And these were all mutations that they published, 150 mutations. Short gastrulation is here. Do we have a known gene? Here we have notch. Extradentical is the cofactor of labial that we have seen. Notch is a ligand. This is just one. Armadillo is a wingless component. These were all mutations. They had funny names. They had everything. Nothing was known about their genetics, about their molecular basis, of course. These were just unidentified mutants, and all you knew is they're on chromosome X, I guess this is, because notch is on the X. Extra-identical as well, so this must be from an X chromosomal screen. They identified in this screen thick vein, pumped, Schnurri, Brinker, and now we know that thick vein is a type 1 receptor, pumped is a type 2 receptor. Good morning, the cat. Schnurri is a component that is very important to repress Brinker, but all of these mutants came from this screen. We just had to find out their molecular nature and then their molecular function. And this has been ongoing for 30 years. I think by now all of these genes are cloned. And so you went from the mutant to the molecule to the mechanisms. And the whole cascade of how a Drosophila embryo develops from, you know that from earlier lectures, you deposit RNA molecules, the mother does that bicoid, you have all kinds of different mutants. So the whole scheme of how you develop an embryo, how do you develop anterior by putting in RNA molecules, these generate gradients, turn on other genes, the interaction of several other genes makes stripes. All of these things came from these genetic screens. All of the parallel genes were identified. One did not know that there are parallel genes. Every other segment lacks. So fantastic genetic analysis in a time when the genome was not known, when none of the genes was known, no sequence was known of any of this. Now, 20 years, 30 years later, we have the full genome. We can ask, are there more genes we don't know of yet? So we mutate them with CRISPR. Maybe they have no function. So now we can make all of this very iterative. We can make 2 mutants, 3 mutants. So this morning when we looked at these 3 days when we looked at signaling, we didn't look at cells, as I said, but what we wanted to do, the aim of the course was to look at cell signaling. How do cells crosstalk to each other? Of course, you can go to a textbook and then you see the pathway. Yeah, that's good. That's at the end what kind of counts. But what we wanted to do, because it's a practical course, we wanted, of course, to show you how you do this. And for this, you need mutants. You collect them yourself. There are so many in the stock collections, you might not have to collect them, but they already exist, and then you order them from the US or from the stock center. You need transgenesis because you want to look at genes, so you need these P elements, and you hopefully know now what a P element is. It's a transposon, and it has been engineered so we can use it, put into transposon whatever we want. A gene with a promoter to see where it's expressed. A gene without a promoter, but a promoter that we can turn on with some other, with GAL4, for example. Anything. You can think about putting in there nanobodies that bind to your favorite protein, anything, but you always use a P element. If you use P elements, you also need balancers because you want to see where your chromosome is. We start to have many chromosomes that we need. We need to generate a fusion protein, fluorescent fusion protein, to see where the protein is. Then we want to manipulate the expression of these proteins. We need maybe a GAL4 line. We need UAS lines. And then we need all of this, the US, the GAL4, the reporter, the manipulator in the same fly. So we need balancers. Otherwise, things become very, very complicated. Or in other words, in a system where you have balancers like flies, you can do very complicated experiments because you can follow your chromosomes. In other systems where you don't have balancers, you have to make sure that the embryo you looked at will have these 3 or 4 constructs. So it's much more complicated. The balancers are of fantastic help. And then we have all these dominant markers that we can use to identify chromosomes. And at the end, we know we put also fluorescent constructs on balancers, so we can see them in the early embryo, not only in the adult via bristles. So I think we have looked at that quite extensively, genetics. So all of these things on how to look at cell-cell signaling can only be done if we have these fabulous genetics, because otherwise we cannot analyze anything. And genetics is not something you are most familiar with, I'm pretty sure, but it's absolutely essential to do these things. It's relatively simple genetics. Of course, we taught you the simple one. There are people out there who do very complicated genetics. So the aim, and that's sort of the take-home message for the exam, we used genetic, and we showed you some of these genetics to understand DPP signaling. So for the exam, what you should know is you should know the DPP signaling pathway. And as always, if you know it all in detail, you'll get a fantastic mark. And if you know the essentials, you will be easily passing. So we don't want to fail you in the exam. The idea is just that you kind of go again through the DPP signaling pathway, and then maybe you remember it a little bit longer in your life, how one of these pathways works. Martin will also ask a genetic question, relatively simple genetic questions. But this is encouragement for tomorrow. Please go to his this lecture where he explains the answers to these genetic questions. So this is what you should learn for the exam, some of these genetics and the components of the DPP signaling pathway. As I said in the beginning, it's the lectures that count. We will not ask you about how you did this or this experiment. There are a lot of this we can go. There are a lot of new things in flies that are very useful now. People start to do disease models much more now in flies. I'm pretty sure that some people work on coronavirus interactions in flies. We have seen also these synthetic receptors from Shinya, synthetic receptor that binds GFP, and so that it has a binder, we put GFP to DPP. This is synthetic biology. This is new, this field. Maybe 8 years old in flies, but it's only emerging now. It took a lot of time to do the first experiments to see how well it works. Proteins can be manipulated directly now, and this is a field that is emerging. So a lot of things also emerge. And you will see more tomorrow about new methods, also about new tools and new approaches, and how proteins of medical importance can be studied in flies. You will see some of these in the tutorial tomorrow. So with this, we go to vertebrates, because Drosophila is not a vertebrate. It has an exoskeleton and not bones. And this is evolutionary-wise a very important question. Where do these genes come from that either make exoskeleton or bones? Do flies have genes that make bones? And actually they do. So this is evolutionary-wise very interesting. All the other organs, liver, kidney, heart, they have precursors in flies. The fly has a liver, it has a kidney, it's the Malpighian tubules. Gustavo showed you these. Fly have a simple heart, it's a linear heart, but this heart pumps. It doesn't have a vasculature, so it's a heart that just pumps in. Now we go to zebrafish, which is also a vertebrate. Few labs in Switzerland work with fish. There are more and more. So Alex Scheer, our director of the Biocenter, he's one of the leading figures in the zebrafish field. I will not say much more. Henry can take over now, so we move from flies to fish. And I hope you enjoy this day, because it's probably the only day in your life you will be able to look at movies of fish embryos and things like that. The other labs, Alex does not participate in the block course yet, so you will not see all that much. Are there questions, maybe to the exam? I don't see anybody now at the moment. Henry is still there. Henry?
Hello. Hello.
Do you have any questions, Henry?
Very informative.
Thank you. So if there are no questions, if there are questions emerging, write to me, but I think it's relatively clear. So I'll give over to Henry And I would like to thank all the fly people so far who have participated. As I heard Gustavo's, what was the name again? Gustavo's explanations were very well appreciated. And I would like to thank him. Also Martin, of course, for all the efforts. And you for being invisibly there. Thank you very much. So Henry, it's...
Yep. Hi everyone, I'm Henry, Heinz-Georg Belting. If you don't mind, if some of you can turn on the camera, then I have the feeling I talk to actually not to myself, but that's very nice. And if you can manage to look modestly interested, that would be even nicer. Some facial response or something like that. You can also shake your head. That's also helpful. Daniel, are you there?
Yes, I'm here.
Okay. So Daniel will sort of, there's a chat function in the down at the bottom of the window. If you want to chat, you can chat among yourself. I'm not going to watch the chat, but Daniel will watch it, and maybe he will then pass on the questions to me by talking. I won't see everyone. I will see like 6 people, I guess, while I'm giving my lecture. And so then I can at least look at the screen and see some faces. That's actually, I appreciate that, as long as it works and the connection gets bad. So a few words about myself. I work with Markus now for 15 years. And actually, he hired me as a zebrafish expert because I did my postdoc using zebrafish in the lab of Wolfgang Griever. And before that, I studied molecular biology, looking at transcription factors. Then I got interested in developmental biology and did my PhD with Frank Ruddle, working on Hox genes. And then I actually moved into evolution a little bit for my PhD, looking at different expression pattern of Hox genes and their regulation in different species. And then I thought I have to do something serious, so I wanted to do something where I can actually look at the embryos and manipulate the embryos. So I thought I moved to zebrafish. And so I start my lecture now, and I hope this is gonna work. Hold on a second. No, that doesn't work, so I have to share the screen first. Okay. How are things?
Beautiful.
Okay, so I see only 3 people, so you can take a turn. So I see Daniel and Markus, and the others I cannot see while I talk. So you can as well turn your cameras off, or you can.
No, don't do that. Don't do that.
Or you look at each other in the meantime. Maybe that's also helpful. Okay, today I give an introduction into zebrafish development. So we have this lecture now, and immediately afterwards we have sort of a virtual practical part. And yeah, so in developmental biology, the major question, of course, is, and you may have asked that question yourself, where did I come from and where will I go to? Well, in developmental biology, we ask the question, where do we come from and how did we become what we are today in a physiological, anatomical sense, and not spiritual. So the question is, when you get from the egg, this is a single cell embryo, to a full-formed organism, for example, how are the body axes specified? How do you make different cell types in the different organs? And how do you actually make an organ? How do the cells manage to associate, find each other, and actually build structures out of themselves? So today this part has, there's two parts. This morning we do live observations of early development. So we look at embryos and discuss what happens at different stages of embryogenesis. And in the afternoon, we look a little later at organogenesis, and our organ of choice is the cardiovascular system. So we're going to look at different aspects of how blood vessels are formed and how the blood vessels form a network and how the whole circulatory system is being generated in the embryo. And now the question is, I better skip this slide. This is basically back to my introduction. Anyway, I tell you about this. So when I studied in the 80s as a student in Cologne and later in New Haven as a PhD student, we had really bad textbooks. So the good textbooks that you have today, like the Wolpert and the Albert, they appeared in the 80s. Before that, it was all black and white pictures in the developmental biology books. And as Markus described earlier, people were looking at adult structures. There were some people that looked at mechanisms of development. And so you got like vertebrate development would look like this early on. People would make manipulation transplantations and look at the structures that are formed. But there was no genetics in the beginning. And so people could study developmental biology in a very classical sense, which I thought was at that time all you could do, but it was not a very attractive field to be in until the early 80s. And on the other hand, since we had molecular biology because of restriction enzymes and people were studying protein DNA interactions very extensively, molecular biology and developmental biology were separated fields. And that changed in the 80s when people discovered the Hox genes and suddenly could now connect molecular biology, the work of how do transcription factors work, and integrate that into a functional network of how actually these gene interactions, this molecular biology was responsible to actually forming these organisms. So there was a fusion of the two fields in a way, of molecular biology and developmental biology. And so that's how molecular biology started joining the field of developmental biology. The problem at that time was in the early 90s that there was a lot of people who were very good with molecular biology. They could clone, they could do a lot of things. So one of the big things to do in mammalian development, because there was not a lot of people that did mammalian development. Different story for Drosophila, because these people were very well trained in developmental biology. But then there was a molecular biologist and said, now we do molecular biology on mammalian development, but they were pretty clueless. They had no idea about how structures were forming, how the embryo is actually made. There was like maybe 3 or 4, maybe half a dozen good developmental biologists that could actually understand what was going on during mouse embryogenesis. Most other people were just looking at reporter genes and looking very meaningful when they looked at the expression patterns of their Laxi mice and being pretty clueless otherwise. So it is very important that when you have a question that you want to study, that you use the right organism. So in the early days, when you look at early embryogenesis, the mouse was not a very good system to study early development because it develops in the mother. It develops very slowly. You can't do much with it other than doing antibody stainings and immunohistochemistry and in situ hybridization. So there's different model systems. You heard about these. These are the invertebrates on the left, the mammals. The chick or the quail is interesting because they're in the egg. You can actually get to the embryo and actually, inject a few cells with something and actually look at cells, how they behave during development, and of course, xenopause. And then now we also talk about the fish. And what I want to explain this morning a little bit is why and how we use fish in developmental biology, and give you a little bit of the history of it too. This is a quiz I will not discuss today, but you can download this. And the idea is that during coffee break or over lunch, you can look at these different animals and put them into the right spot. And the point I want to make is that all these things are chordates, except for one, which is considered here the out group. And then you can wonder about which animal fits what. And I want to give you a sense of the timeline when certain species evolved during vertebrate phylogeny. And we can discuss this a little bit later in the afternoon. Okay, and so the fish was an unusual model until the 90s, and the mammals are 220 million years old, and that's one thing I always, get a little bit concerned is if people talk about fish as being primitive vertebrates, that mammals are more advanced than fish. I mean, evolution happens in parallel. There is no really primitive and advanced organism in the sense, because all the organisms that we have are adapted to what they need to do. And so they had evolved different strategy for survival, but in principle, cyprinids, for example, are more modern than mammals if you want, because mammals already existed 220 million years ago, but cyprinids only 60 million years ago. And also, the modern fish are 50% of all living vertebrate species are teleosts. And you see that there has been a huge radiation in modern times over the last 60 million years, when there was a lot of fish that evolved here. And one question is, how could they evolve into so many different species? And what is the genetic basis for that? Here you see this huge radiation of fish over the last 60 million years here. And fish are very heterogeneous. You see that in different shapes and sizes and how they adapted to different environments. And so here, these, this is where the zebrafish are. So this is a relative basic fish here among the teleosts. And this is, most of all know this fish. So I would ask, who doesn't know what this fish is? And most people wouldn't know, but most of you have eaten it. This is eagly. It's called percomorpha. I don't know what that is so it's a barsch. So percomorpha, so the perks are the barsche. And so there is quite an evolutionary distance here between those. So this is actually, this radiation of the percomorpha is even more recent than that of most of the other fish. And these are the fish that you famous when you look at the work of Walter Salzburger on the cichlids. So the cichlids also belong to this group of fish. So all this radiation is thought to have been possible to change in genetic programs. And if you change genetic programs, the easiest way to do it is to add more information. And during vertebrate evolution, what happened to get more information is that you have genome duplications. because the genome duplications give you more material to play with, so the genetic programs that actually form these organisms have more, let's say, Lego pieces to play with. And then you can refunctionalize genes from function A to function B. And this is actually what happened in teleosts, which zebrafish is a teleost. And so during duplication, you originally duplicate the whole genome, and then there's different ways how this genetic program can be modified. What you can have is that, for example, here you have two clusters of genes, and the organism keeps only a single copy of each by losing the other copy selectively, and that would basically not change much of the program. But you could also keep one cluster and have the new copies acquire new functions. Or you could just have degeneration and gene loss. So this is a way that we think about how at the genome level evolution may have occurred in different branches of the vertebrate phyla. So these are now the three major branches of vertebrates. So, and they're all fish. So you have Latimeria, which is in German Quastenflosser. You have a teleost here, which is the pike perch. And you have, as an outgroup here, we have the rayfin, the cartilaginous fish, which is a shark, basically. And actually, when you lift the screen, then you see that the coelacanth actually groups together with the mammals and all the other vertebrates. So what we have here in this clade was, we have three major branches. One with the coelacanth, with this fish, and this fish belongs basically to humans, and the humans are more closely related to coelacanth and vice versa than the coelacanth to the zebrafish. Just to give you a sense of how evolution worked, that we actually, in a strict sense, we would belong to these fish and not to these fish. Nevertheless, besides this 200 million years of separation between coelacanth and zebrafish and mammals, there's a high degree of conservation, which is quite obvious, but also you can trace that back to the genome level. And I find that quite remarkable. There has been, I think, even 400 million years of separation. And if you then go to the chromosomes and look at the genes and compare the genome between humans and fish, you will find that on zebrafish chromosome 3 here, you will find that in human chromosome 17, there is a lot of stretches here in the zebrafish chromosome 3 that correspond actually to human chromosome 17. So it's the same genes in the same order. They have kept their arrangements. And so there has been some shuffling, but it really shows the basic plan in vertebrates on the chromosome level in the gene organization has been conserved. And it really shows that we go back to the same roots. So when you look at anatomy, you know, you have a head, you have arms, you have, you know, what Markus also alluded to, the organs are there. So the organs are there, the anatomy is there, the physiology is there. You can have diabetes in fish. You can use it for all kinds of disease model. The gene composition, as I showed you before on the right, also the same. And gene expression patterns and function, they're all conserved. So we can use a fish as a model organism to look at general vertebrate development. To illustrate it a little bit more, if we look at anatomy, this is the circulation of a zebrafish and the blood vessels in a mouse embryo. And you see there's quite a bit of conservation here We have here these blood vessels here at the future mouth, and the same is here. We see this nice metameric organization of blood vessels along the body axis, and you see the same vessels here in the mouse embryo. These are called the intersegmental vessels. We will see those in the afternoon when we look at blood vessel development in the fish, and also the cells in the fish and the mouse express the same genes. For example, blood cells express this transcription factor called GATA1, and endothelial cells express this transcription factor SCL. Zebrafish and mouse the same, and in the heart you find expression of NKX 2.5 in the fish and also in the mouse. I just want to walk you through a few of the basic anatomical features that are particular to fish, and everyone knows what a fish is, what it looks like, but it's also good to understand what are actually the specific features of a fish. So one thing that is obvious is that you have fins, and the fins, we don't have fins, but some of the fins are actually homologous to some of the structures that we have, and those are particularly the pectoral fin that is homologous to our arms. So it expresses basically the same genes as our arms, and they're formed in a similar way, and the pelvic fin, which corresponds to our legs. So in a way, they also have these, the four members. They have forearms and legs, but they don't have hands. So tetrapods means having four hands. The fish don't have hands. So they have everything like the upper arm and the underarm, upper leg, lower leg, but no hands and no feet. They don't have lungs, but they have gills. So they take up the water, and in the gills, they take up the oxygen, and they get rid of the CO2. And they also get rid of ammonia, which is a metabolic product from, for example, protein metabolism. And that can be very toxic if it becomes ammonia. So this is ammonia, so ammoniac. So this is ammonia here. They have a swim bladder. We have lungs, they have swim bladder. And what is interesting to see here in the zebrafish, the swim bladder is connected to the esophagus, which is the foregut, and they can actually fill the swim bladder by swallowing air, and there's a connection between the intestinal tract and the swim bladder. And actually, the fact that they can swallow air and fill it is actually quite an analogous function between the swim bladder and the lung. Something happened here with my presentation. So I give you a few facts about the zebrafish, what's important for us in the lab. And so where does it come from? It comes from India and from Bangladesh. It lives in all kinds of different environments. It can live in rice paddies. It can live in creeks. Mostly it's slow-moving water, not fast-running rivers. They are 4 to 5 centimeters big when they're adult, and they reach sexual maturity when they're 3 months old. So you basically can have 4 generations of fish per year. However, and that is a great advantage of the fish, you can collect eggs every week or every 2 weeks. So the cycle of oogenesis is about 1 week. And you don't have to kill the mother. So if you take embryos from a mouse, you have to kill the mother. Here, we just collect the eggs and let the mother live. So that is a huge advantage. The lifespan in the lab is 2 years. In the wild, it's usually less than 2 years, but they can also get 4 years old. We hate to see that in the lab. When fish get old, they start, dispensing all of their diseases and dying, who you can all see I always tell him to kill his fish when they get too old. They have 25 chromosomes. They have a DNA content of I think that is in American English. That's American. It's trillion. So in Europe we say billion. But the genome size is basically the same as humans, maybe half the nucleotides. But actually, they do have more genes because of their genome duplication that occur in teleost. Fish have more genes than mammals. There's a lot of labs in the world, and now also in Switzerland, it's about 20 labs working with zebrafish. So when we started 15 years ago in Basel, there was only 2 or 3 labs in Switzerland working with fish, and now there's more than 20. And the tendency is increasing. I'm running a little bit out of time, so I have to speed up. So the guy who came up with this idea, George Streisinger, started zebrafish research because he thought he wanted to do things he wanted another genetic model than mouse, and he wanted to have a system, a vertebrate system, where you could actually do genetics, and that meaning in the sense forward genetics. And so he played around a lot, and he was able to actually make clonal zebrafish. And that is actually the first time that zebrafish research hit the headlines in Science and also in the newspapers, that people were able to actually make clones of a vertebrate. And that was in the 1980s. And I skipped these things here. So why were people interested in using zebrafish as a model organism? Well, one thing is we wanted to use a vertebrate organism, and where you could do forward genetics and do also embryonic manipulations. And these models here, they were good for different things. With mouse, you can do reverse genetics, and it's a mammal, so it's interesting. With chick or quail, people were able to do embryonic manipulations, but there was no genetics available. And generation time is very long. And also with xenopus, you can't really do genetics. And so with zebrafish, there was a model now with the promise of being able to do forward genetics. Now we can do also reverse genetics. You can do embryonic manipulations, and that means, for example, you can inject substances in the fish, you can do transplantation experiments, you can put fish from different individuals into one, so to make a chimera. Then it's also very practical. It is small in size, you can put the embryo under the microscope. It has a short generation time, as I mentioned, so that is important. If you do genetics, you have to, breed different alleles of different mutations or transgenics together, and you get a high number of progeny. You don't have to kill the mother because of external development, and you can watch it from fertilization to death, basically. The breakthrough really came for the fish when actually 2 labs succeeded in doing 2 large-scale screens to look for genes that are important in embryonic development, and these are forward genetic screens. This is a schematic on the left, how this is done. This is not important for you. It just want to mention this goes from a mutagenesis of a male to the actual screening and isolation of genes through 4 generations. So you have a parental generation, and then you have so the grandchildren of these fish here. This is where you actually screen for your mutants. So this takes at least 1 year, but because the screening takes a long time, a screen like that screen took about 3 years to do, to find several hundreds of genes that they could identify to be important for vertebrate embryonic development and isolate the mutants for these genes. So this was done in Boston in the lab of Wolfgang Drever, and also, as before, the screen for Drosophila was done also. There was a zebrafish screen in Janni Nüsslands-Vollart's lab in Tübingen. And these two screens were published in a single issue of Development in 1996. And that was, I think, the thickest volume they ever had. It was about 40 or 50 articles on these mutations that they found in the zebrafish. This is redundant to what I told you before. So, salient features of the zebrafish model. And here, okay, also the title. So what I want to bring home, this is important, that we do not work on fish because we are just, fish aficionados and in love with fish. We think it's a great model to do what we want to do. And so we choose this model organism, the zebrafish, because of our research objectives and the experimental design. And these are the features of the fish that allow us to do these particular experiments we want to do. And that is following, for example, vascular morphogenesis and being able to do time-lapse and do genetics. And these are the reasons why we can do this. We can do live imaging because it develops outside the mother. It is very fast. You can basically look at the full development from an egg to a vertebrate embryo that looks like a vertebrate that has all the parts in one day. They are transparent. We can label all the cells. We can follow single cells in development. We can label them and then follow them. They are small, so we can look at the whole organism in one go. High number of progeny allows us genetics, so we can do forward genetics. And now, because of CRISPR and other methods, we can also do reverse genetics, and we can do manipulations, for example, maker mirrors by cell transplantation. And of course, as I mentioned, fast development, transparent embryos, and small size of embryos, that's for us in the Affolter lab, the great reasons why we do use the zebrafish, because we want to understand organ morphogenesis. I want to show you a few examples of some movies, is just show you the strengths of the model system. And there's 4 videos, one dealing with early morphogenesis, neurobiology, cell biology signaling, and the last one is an example from our lab, showing cell-cell interactions during blood vessel formation. So this all, what we do today in the morning, happens between 30 minutes and 1 day of development. And this is, you can put a single embryo under the microscope and you can watch all this under the microscope, basically overnight and next day. This is what the embryo looks like on the left. This is a 24-hour embryo. So this embryo is 24 hours old since fertilization. You see it has a head, it's the brain parts here, it has a yolk, and this is the trunk and the tail. And here on the right-hand side, it was an embryonic manipulation. People have transplanted cells into the embryo, and they now follow here in the tail bud how cells are actually integrated into the different tissues. And that is shown here. So this shows you respective cells in the tail behaving here in the tail bud. And actually, then you have these 2 cells here in the notochord. And in this video, these cells have been marked. So you see cells at the tip of the tail, and then these cells become integrated into the notochord. That shows basically that in the tail bud, there's a mixed population of cells, and some of them become incorporated in the notochord and other cells don't. These are a few movies showing a reporter for intracellular calcium release. Intracellular calcium release is important in in intracellular signal transmission in the central nervous system. And this is expressed now in the midbrain of the fish. And the dorsal part of the midbrain is called the tectum, and this is called the optic tectum. And this is where, visual input is being processed. So there's this fish, he looks at a paramecium swimming by. It's food for the fish. They like to eat it. And you see that different parts of the brains become activated depending on where the paramecium is swimming. And thereby, we can try to understand how the neuronal networks work, and the pathway between the eye and from the visual input into how this is processed in the brain in the end. There is a lot of fancy tools where we can activate pathways in individual cells. This is called optogenetics, which means we have a laser and then we can actually activate a pathway, and then we can look at the response of cells to this activation of this pathway. And what was done here was we had a laser that can activate the PI3 kinase pathway. And what this pathway does, it's important for the formation of lamellopodia in front of the cell. So this pathway gives the cell orientation and tells the cell where to go to, because where this pathway is activated, this cell will form a lamellopodium, and this is where the cell is going to go. And actually, you can use this laser now to actually guide the cells through the embryo, and that is shown in the next video, where you have, these are three examples. You have one cell here, one cell here, and one cell here. And these cells now become stimulated by a laser beam, activating this PI3 kinase pathway, and look what happens. You can make them migrate where you want in a way that they can actually spell the word RAG. And RAG1 is one of these big regulators of PI3 kinase, downstream of PI3 kinase. So this is a thing we can do in fish. And then in our lab, we are interested in how three-dimensional structures are formed, how organs are formed, how cells can build a vasculature, and what the cells are actually doing during this process. It's built on cell-cell interactions, so these are very important. And this is very complex. We try to separate these into different processes, but this is a kind of analysis that we do in the Affolter lab. Back just a little bit. So what I want to show you now is a movie about how zebrafish is used in biomedical research.
We sequenced the zebrafish genome to a very high standard so that we could really know what the direct comparisons between human genes and zebrafish genes are. I know it seems like an odd choice to study zebrafish, especially when looking at human disease genes. Zebrafish are very easy to maintain in the lab in large numbers. The embryos are essentially transparent, and that means that you can see every cell as the embryos develop. The genome is extremely similar. So 70% of human genes have a zebrafish counterpart. And if you look at the disease genes in human, what we find is that 84% of disease genes in human have a zebrafish counterpart. So for example, the greatest cause of muscular dystrophy in humans are mutations in a gene called dystrophin. And zebrafish have a dystrophin gene. It's very similar. Mutations in the zebrafish dystrophin gene give zebrafish muscular dystrophy as well. We developed this zebrafish mutation resource funded by the Wellcome Trust to help scientists and doctors understand how gene variation affects inheritance of disease and susceptibility to disease. So, for example, we can construct a mutation in zebrafish that's very similar to the human mutation, and then use the zebrafish as a tool for screening for new drugs. The goal of biomedical research in general is to improve human health, and we hope to improve human health by studying zebrafish genes.
Okay, I show you another movie just to show you what life in the fish house is like. And this shows you just some fish tanks, and I will comment on this video as I play it. What happened here? I have to go back. Yeah. So fish are swimming around in big tanks of about 10, 12 liters. We can keep 50 fish in one of these glass tanks. But there's also smaller tanks where we keep fewer fish, between 10 and 15 fish. They are being fed. And then we set up matings. And the most important things for setting up matings is to distinguish between males and females. This is a female. And you see the male on the bottom has a smaller belly than the female. For some reason, this presentation is playing a lot faster than it's supposed to do. But in the end, never mind. What is going on here? Okay. Something. Not good. Okay, I'll show you this video again. The fish look rather nervous because the movie is playing too fast, and I can't do anything against that. My computer is acting up. So we are feeding the fish with Artemia. We breed the fish, so you have to stay with me through this ordeal. But it's nice to look at fish anyway. So you have a male, the smaller one is the male, it looks a bit more orange. And this is a female, it has a nice plump belly where the eggs are. So the male has a smaller, oops. Yeah.
Henry?
Yes?
Maybe make the point where the eggs are and what the idea is about the fish not eating their own eggs. And
yes, thank you. So what you saw here, I can't show you this anymore, is they were in a cage, these eggs, where they have a grid. So there's at the bottom here, that's perforated, there's holes in there. So when the female lays eggs, they're fertilized by the male, and actually, the fertilized eggs, they fall through this grid into the bottom. And then what we do is we take the fish out, and then we collect the eggs from the bottom. And this is what the eggs then look like in a petri dish when we have collected them.
otherwise, they would be eaten up by.
Again?
They would be eaten up, the eggs.
They would be eaten up. Yeah, they're carnivores. They like caviar.
Exactly.
And actually, you're not supposed to feed the fish with eggs because there can be diseases transmitted by the eggs from fish to fish. So that's not a good thing. I think there's some people in the past, not in our lab, but other labs that have fed spare eggs back to the parents because it's good food, but actually they can recontaminate themselves. It's a bit like coronavirus. Okay, a few more slides left. Oh shit. Hold on a second.
So let me use this second turn on my camera. Because,
Okay.
Henry,
Yes, I'm back.
I take up these 2 minutes.
Yeah.
Because there's something important we should discuss. Is this how we do these presentations tomorrow? The idea was initially that we had, you came back too fast, Henry. So I tried to fill the gap. Shall I quickly say that now, or do you want to finish?
Yes, yeah.
Okay. So the plan was that every group presents to the whole community 45 minutes, 30 minutes. And then we have 3 presentations in the morning, 3 in the afternoon, and you have heard about all these papers, which is good, but it's a big group. The suggestion was that we do it in smaller groups, and we can do that and take a bit more time. Then you can discuss more, and you do it among yourselves. So here is my suggestion, and then we can either do it as planned or do it as this new suggestion. If you have another one, that's okay, but we have to make up our mind today. My suggestion is that in the morning, we always pair 2 groups, and then they have, instead of 30, they have 45 minutes to go through the papers. Group 1 presents to group 2. Then you can make a break if you want. You organize it as you want. And then group 2 presents to group 1. In the afternoon, however, and then you're not done because we have another session in the afternoon. In the afternoon, group 1 would have to present again to group 4. And group 4 would present to group 1. Same scenario. You have twice 45 minutes. You can take 2 times 30. You can do what you want. And then you are in smaller group and you can discuss. But you're still 12, 6 plus 6 usually. But this is my proposal. Is there anything somebody wants to say? You can maybe discuss it over lunch or anytime. I think it's either one of these. The advantage is you're a smaller group. The disadvantage, you have to present the paper twice. But if you do it once, you can do it twice. If you want, you can record it and play it in the afternoon. You can organize your session the way you want. The one who suggested that we discuss that, who was that? Are you here?
Yes, that was me.
You caused all this trouble. I don't need to know the answer now. Just come back to me. If you have another suggestion, it's okay, but what I think is not good is that you say, okay, let's just do one session and then we are done, and then I don't think this was the idea anyhow. The idea is
but
safe. Yeah.
I'm just not sure if it will work to really discuss for about 3 hours, because that's quite intensive, and I don't know if people will have the energy for that.
You're our top students in Switzerland at the top institute. I'm sure you can do 3 hours. I mean, you listen to the paper and then you discuss five minutes. I think this is not such a big deal because you present 6 different people. So you listen for 25 minutes and then you can discuss and take it easy to discussion. If you do it this way, you can take a bit more time. I would prefer the second scheme, but you have to present twice. If you want, you can record it the first time and then play it in the afternoon the second time and not do it again. I don't know how, we tried it, it seems to work. Think about it, discuss it, and get back to me. It's just for you that you know that it doesn't change anything. The presentation is 25 minutes. I think this is a good time to have everything done. It's no use to present only 10 minutes, then you don't go in detail. Present much longer is also not a good idea, I think. Also, I think we don't want to change that because then you don't know what to do. Okay?
Okay.
You can write me. We can also call me back at some point today so we can finalize so that you all know what you have to do. But it doesn't change the preparation of the presentation. It's just the modus that would change. Okay?
Yes.
Henry?
Yeah.
Sorry for...
No, that's fine. No problem. Hold on a sec. So okay. Everything good? You see it? So this is basically now this morning's program.
You have to share your screen, Henry.
Okay. Thank you. Fuck. Hold on. Okay, now. That works?
Yeah.
Okay. So now you got these eggs. In principle, virtually, you have your eggs now. We gave them to you. You got embryos in different stages. They're all now, instead of a dish, they're already in a video. I hope you downloaded the videos to your desktop. If you haven't done that yet, you can do that in the break that is to come. And Janmin, who can't be with us today, he made these videos, and what we do in the afternoon is we look at these videos, in the morning we look at these videos together and trying to state the obvious. We walk you through these videos, and then we discuss the different stages of development, what happens where and when. And so in the past, we would make drawings. You don't have to make drawings. It doesn't make sense in this setting because it's difficult to do. In the past, we would do it like this, that you take this embryo on the left, you see it on the microscope, and then you would just point out the most essential features that you see in the embryo, something like this. We don't do that anymore, so we do it now all online. So what you need to have ready on your desktop or on your laptop at home anyway are the videos and the part A drawings. That's a PDF that is helpful for the discussion. So you should have those things open side by side. We are distributed over 4 groups. You get an invitation either from Daniel, from that's Daniel here, from Katrin, from Maria, or from Alessandra. I do it jointly with Alessandra, group number 4. And then I would think we take a 10-minute break where you can make yourself a coffee.
15.
And then we start at 10:20, let's say 10:25.
Can we make a 15-minute break, Henry?
What?
Can we make a 15-minute break?
Why don't we make it 10:30 then? Let's get together at 10:30.
That's so you go from 10 to 20.
Yeah, we make it. Yeah, 10:30 is a good time to start.
Okay.
Is everyone okay with that? Do I hear any objections? I don't hear any objections.
Can we say that we meet directly in the Zoom groups that they have been invited at 10:30?
Yes, yes. At 10:30, you go directly to the Zoom group. The 20 minutes also gives you a few minutes if you haven't downloaded everything that you need for now, because it's a big package of movies. It's several hundred megabytes. Maybe it's so. I hope that will all work.
And then we stay in the groups and discuss the movies until 12.
Until 12:30, I would say. The program says. What does the program say?
Program says 12.
Yeah, but that doesn't. I mean, I knew already that was too short. I mean, if you do 12:30, hold on a second. I mean, we don't have to, so no, that's not it.
Probably each group will finish at a different time anyhow.
Yeah, yeah. We're done when we're done. So it will maybe a little later than 12. Let's say it could be 12:15 or something like that. It's just that these are the 10:30, and then we finished 12:15, something like that. Whatever it takes.
So if ever you want to get back to me for tomorrow, write me an email, and then we organize via Zoom or via something. Enjoy your coffee.
Okay. See ya.
Bye.